Hello and welcome to our virtual CYTO 2020 tutorial. Today we'll be talking about calibration methods for small particles. My name's Joshua Welsh and I'm a postdoctoral fellow at the National Cancer Institute. I'm joined today by John Nolan from the Sint-Allon Institute and Edwin van der Pol from the Amsterdam Medical Centre. So the plan for today is to give a brief background to calibration, what it is and why it's useful. This is then going to be followed on by a demonstration of how to use some of the currently available calibration tools. The remainder of this session will then be used to answer any questions you might have. So to set the scene, small particle detection is a growing area of interest, and over time we have seen that the concentration of EVs has continued to increase in the published literature. This is likely due to increases in sensitivity as well as optimization of assays. The real problem though is that no flow cytometer is capable of detecting the full range of EVs. This means that the reported concentration in the literature is not reliable and it's hard to validate without knowing the limits of detection for these studies. So earlier this year, a position paper was published in the Journal of Extracellular Vesicles. This is a reporting framework that was developed by researchers from around the world representing the ISEV-ISAC-ISTH societies. While the main focus of this work was to provide a standard framework for reporting single EV flow cytometry experiments, a set of minimal reporting requirements was also included. So just to give a quick breakdown of this framework, the first two components are designed to report the assay metadata and include the existing MISEV and MIFlowCyt standards. This also includes some sample preparation steps. The third component focuses on reporting and demonstrating assay controls. So up until this point, if you've completed all of these three components perfectly, you'll have demonstrated that you're able to detect single EVs and you'll have provided enough detail for others to reproduce your assay. You won't, however, have provided enough detail for others to validate your assay. So components 4 and 5 are focused on reporting instrument acquisition and information on calibration and methods for EV characterization. It's these steps that will enable you to derive your limit of detection and report your data in standard units. And this will allow others to validate your assay on instruments that don't have comparative sensitivity. So the final two components, 6 and 7, are mainly focused on data quality control. These include how you decided to gate your data and report statistics, as well as providing your flow cytometry files for others to inspect. So what is calibration? Calibration is the conversion of arbitrary units to standard units. In the top right plot, you can see that if we acquire the same population at 3 acquisition settings on 2 different instruments, we're unable to overlay that population on the arbitrary unit scale. This is where the problem lies when we report EVs that we cannot fully detect and where we have no idea what proportion of the EV population we might be detecting. You can see once we calibrate this fluorescence scale from the arbitrary units to an equivalent soluble fluorophore scale, in this case PE, that regardless of the instrument platform, the optical configuration or the detector settings, we're able to overlay this same population of data and we're able to make fair comparisons. For the majority of flow cytometers, we're only quantifying 2 different signals, the fluorescence signal and the light scatter signal. For fluorescence calibration, we tend to use reference materials that have known quantities of fluorophore on their surface or known antibody binding capacity. This relationship between the number of fluorophores and the intensity in the majority of cases should be linear. In the case of light scatter calibration, the relationship for calibration of diameter to signal intensity is nonlinear. To calibrate this scale, we need reference materials of known size and refractive index. Not only is the relationship between the particles and the light scatter intensity dependent upon the diameter and the refractive index, but it's also dependent upon the optical geometry and collection angle of the system. So this is a more complicated parameter to calibrate when compared to fluorescence calibration, but today we'll demonstrate 2 dedicated software packages that will perform small particle light scatter calibration without needing to know the complex physics required to achieve it. So there are many reasons for why you'd want to perform calibration on a flow cytometer. It will allow you to decide which instrument has the highest sensitivity and resolution, quantify which settings are best for any particular instrument. It'll also allow you to define the limit of sensitivity for your assay and normalize for day-to-day variation. Importantly, it will allow others to validate your assay and lay the groundwork for standardized multi-center studies to become a reality. For the sake of completeness when talking about reference materials and calibration, I wanted to quickly mention validation and quality control reference materials. In the EV field, there is a growing body of literature talking about the need for biological reference materials. While these are going to be useful for developing assays and validating if assays work as positive and negative controls, they're unlikely to be useful as calibrators. Furthermore, the use of beads without quantified units are useful for tracking instrument and assay performance over time as quality control, but are distinctly different from calibration reference materials. Before we go on to demonstrate the calibration tools available, I also wanted to cover concentration as a metric for quantifying EVs. Concentration is a metric that is regularly used in the EV literature and in the vast majority of cases is reported as absolute concentration. We already know, however, that our instruments are not capable of detecting all of our EVs, so this method of reporting doesn't allow us to validate others' findings. For the field to become truly standardized, we need to report the detected number of EVs above a limit of detection, and the way to calculate this limit of detection is going to be by using calibration of whichever parameter the EVs are being enumerated on. Finally, I wanted to demonstrate the utility of calibrating the parameters being used to enumerate small particles. This work was recently published with Vera Tang at the University of Ottawa and was based on a tutorial we gave this time last year at CYTO. The top plots show the detection of GFP-expressing murine leukemia virus, which is approximately 124 nanometers. This was detected at 3 different settings across an LSR Fortessa and CytoFlex-S. You can see that if I draw a representative gate around the murine leukemia virus on the CytoFlex's plot, none of the virus appears in that same gate on the LSR Fortessa. This demonstrates the problem of comparing data using arbitrary units. If, however, we calibrate the fluorescence parameter to molecules of equivalent soluble fluorophore and the light scatter parameter to diameter, you can see that the same gate drawn across both flow cytometers is effective. So now with our calibrated units, when we want to report concentration, we can see that the LSR Fortessa actually wasn't able to detect all of the virus. We were clipping some of the virus due to the side scatter threshold. In this example, the total detected virus at each settings is identified by the black gate, while the red gate takes into account the detectable virus accounting for the limits of detection across the instrument platforms and detector settings. If we look at the detected concentration of virus across the instruments and settings using the black gate, we can see the concentration is higher on the CytoFlex-S, which is expected. If, however, we look at the concentration of detected virus when constraining the gate to the limits of detection across both platforms and settings, which is the red gate, we obtain comparable data. So this is a great demonstration of why reporting concentration above a set limit of detection is going to help standardize our field. It allows others to validate our work even if their instrument differs in sensitivity. So I'm now going to hand over to Edwin van der Pol, who's going to take you through the Rosetta Calibration software.
My name is Edwin van der Pol and I will introduce Rosetta Calibration. I have one conflict of interest to declare. I am a co-founder and shareholder of the company Cytometry. Rosetta Calibration is a kit which turns your flow cytometer into a particle size analyzer. In this workshop, I would like to discuss the relevance of particle sizing, and I would like to discuss the operation principle and the practical implementation of Rosetta Calibration. In addition, I would like to explain how we validated Rosetta Calibration and what the pros and the cons of the approach are. This graph shows the concentration versus diameter of extracellular vesicles and other particles found in urine. Please note that the vertical scale is a logarithmic scale and that right from the peak the distribution follows a power law function. So if we have two flow cytometers, one flow cytometer with a limit of detection of 180 nanometers and a second flow cytometer with a limit of detection of 230 nanometers, they measure already a twofold difference in concentration in this sample just due to differences in the limit of detection. So for data comparison, it's important to measure particle concentrations within the same size range. Therefore, it's bad practice to state we found 200 million particles per milliliter. Good practice would be to state we found 200 million particles per milliliter ranging 180 to 600 nanometer in diameter. Here you can see the side scatter versus forward scatter of extracellular vesicles and other particles found in human urine. Please note that these axes have arbitrary units, and therefore it's not possible to confirm whether the detected particles are within the expected size range. So also for data interpretation, size does matter. Thus, Size does matter for the detection of submicrometer particles to enable data comparison and data interpretation. Regarding data comparison, I don't only think about comparing data to other labs, but also to correct for day-to-day variation and to allow other labs to reproduce your data. Regarding data interpretation, you don't only have access to an important sample property, such as the diameter of your particle, but in turn quantify your sensitivity. And you can use that, for example, to optimize instrument settings. So how does Rosetta Calibration determine the particle size? The first step is to measure the light scattering of beads. So here you can see the side scatter versus diameter of 6 populations of polystyrene beads measured with a bead defects caliber. So please note that again the vertical scale is a logarithmic scale. As expected, the scatter intensity increases with increasing diameter. However, this is not a calibration, as we don't know what happens in between these data points. In order to know, we have to describe these data points with a theory. Rosetta calibration is based on MIFlowCyt-EV. When the particle diameter and refractive index are known, and when the laser wavelength, polarization state and power are known, MIFlowCyt-EV can be used to calculate how much light a particle scatters in each direction. Now, if we integrate the scattering function over the collection angles of the flow cytometer, we get a theoretical description of the scatter to diameter relationship. As an example, you can see here the collection angles of a forward scatter and side scatter detector in an apogee flow cytometer. After measuring the beads and after applying theory, we get the scatter to diameter relationship. As you can see, the theory describes the data well, at least for polystyrene beads, which have a refractive index of 1.605. To show that this procedure also works for particles with a lower refractive index, we applied the same procedure to silica beads, only changing the refractive index in the model to 1.445. As you can see, the model describes also the data of silica beads well. Now we have calibrated our instrument using polystyrene beads and MIFlowCyt-EV and validated this approach using silica beads. We can use this calibration to predict what would happen with extracellular vesicles, or EVs, as you can see here in green. So we calculated here the scattering to diameter relationship of EVs, assuming that EVs are a core shell structure, where the shell is 10 nanometers thick, has a refractive index of 1.48, and the core a refractive index ranging from 1.36 to 1.40. As you can see, the light scattering of EVs is considerably lower than silica of polystyrene beads of the same size. So from this we have learned that with this flow cytometer, when measuring 500 nanometer and 900 nanometer polystyrene beads, for example to gate EVs, you select rather particles larger than 2 micrometers. In addition, we now know that because the detection limit is close to this 200 nanometer silica bead, that corresponds to the detection of about 350 to 600 nanometer EVs. So we quantified the sensitivity of our flow cytometer in terms of EV diameter. So how does this work in practice? Do you have to be bothered with MIFlowCyt-EV? In practice, I suppose you measure light scattering and analyze the signals with your favorite flow cytometry data analysis software. As an example, I showed here the side scatter histogram of particles from human urine. Because the data are in arbitrary units, this plot provides limited insight. The idea behind Rosetta calibration is to fully automate light scattering calibrations of flow cytometers. The first step is to measure a mixture of polystyrene beads, and then to read the corresponding flow cytometry data file by the software which comes with Rosetta calibration. The result looks something like this. So here you can see the scattering intensity histogram of the bead mixture. And due to this software, you don't have to bother about which peak corresponds to which diameter, and you don't have to think about your flow cytometry type and collection angles. It's all pre-programmed in the software. Thereby, the software is able to relate these bead peaks automatically to their corresponding diameters, and then the software automatically describes the light scattering intensity versus diameter by MIFlowCyt-EV, taking into account the refractive index of polystyrene beads for the wavelength of your flow cytometer. The next step is to model your particles in order to calculate the scatter to diameter relationship for the particles of interest. And once you have done that, you only have to hit export, and the size distribution of your particles is added to the flow cytometry data files automatically. If you now open the processed files of your samples with your favorite flow cytometry data analysis software, you will find a new channel, diameter in nanometers, which allows you to make particle size distributions. To validate Rosetta Calibration, we have performed an interlaboratory comparison study, which was sponsored by the International Society on Thrombosis and Hemostasis. We validated the approach on 46 different flow cytometers. With Rosetta Calibration, 391 nanometer silica beads were sized 373 nanometer with a CV of 5.8%. 772 nanometer silica beads were sized spot on with a CV as low as 3.7%. In addition, we investigated the variability of the measured EV concentration using different approaches. We found a CV of 139% with the previous standard, which was based on a gate of polystyrene beads. By expressing the concentration of EVs within a calibrated size range, Rosetta Calibration reduced the CV to 81%. This dazzling slide shows the scatter to diameter relation for 15 flow cytometers in the interlaboratory comparison study. I prepared this slide to demonstrate that the scatter to diameter relation differs per flow cytometer and to convince you that calibration is important. To quantify the sizing accuracy and precision, we have measured the size distributions of a mixture of polystyrene beads with nanoparticle tracking analysis, resistive pulse sensing, and flow cytometry followed by Rosetta Calibration, and fitted the results with Gaussian functions. Please note that the bead concentration decreases with increasing diameter, like in many biofluids. It is clear that nanoparticle tracking analysis measured the broadest peaks, whereas flow cytometry measured the narrowest peaks, indicating that for sizing beads, nanoparticle tracking analysis is least precise and flow cytometry is most precise. To investigate this further, we quantified the relative sizing error and the measured coefficient of variation of the beads. As a reference, we added the data obtained from a NIST traceable electron microscope. The closer the data points are to the black line, the more accurate the method, whereas the smaller the error bars, the more precise the method. From the results, it is clear that resistive pulse sensing and flow cytometry achieve the best accuracy in sizing beads, and that flow cytometry is the most precise method for sizing beads. As a proof of the pudding, we measured the size distribution and concentration of particles in urine with the same detection methods. Resistive pulse sensing measured the smallest particles and therefore the highest concentration, followed by flow cytometry and nanoparticle tracking analysis. For particles exceeding the detection limit, flow cytometry closely follows resistive pulse sensing. Above 200 nanometer, nanoparticle tracking analysis also follows resistive pulse sensing and flow cytometry. However, below 200 nanometers, nanoparticle tracking analysis underestimates the particle concentration, as confirmed previously by Philip et al. And Gross et al. For other polydispersed samples. All in all, we can conclude that light scattering signals of flow cytometers can be used for sizing submicrometer particles and generally lead to higher accuracy and precision than nanoparticle tracking analysis. The pros of Rosetta Calibration are that you can measure an absolute particle size distribution with number concentration per milliliter on the vertical axis. In addition, with a measurement error smaller than 5% and a CV smaller than 4%, flow cytometry-based sizing is accurate and precise, especially compared to nanoparticle tracking analysis. Because flow cytometry has high throughput, A size distribution based on hundred thousands of particles can be measured within 2 minutes, thereby guaranteeing statistical significance. Because flow cytometers are getting increasingly more sensitive, sizing of EVs, even smaller than 100 nanometer, is nowadays feasible. Finally, the approach is widely applicable, as flow cytometers are widely available in hospitals and laboratories. In turn, also fluorescence labeling and fluorescence triggering is no limitation to Rosetta calibration. The cons of Rosetta calibration are that the model assumes spherical or concentric particles with a similar, known, and isotropic refractive index distribution. In plasma, all EVs smaller than 500 nanometer are spherical, so for plasma EVs the first criterion is met. For EVs, more measurements of the refractive index distribution are required to validate whether a core shell model is suitable to describe all EVs in a biofluid. Moreover, Rosetta calibration currently assumes isotropic scattering of the membrane, whereas in reality, the membrane may scatter light anisotropically, because the phospholipid membrane of EVs have different refractive indices in the radial and transverse directions. More research and measurements are needed to confirm anisotropic light scattering of EVs. Another disadvantage is that the scatter-to-diameter relationship does not always have a unique solution. On the right, you can see that the forward scatter light versus diameter has no unique solution for polystyrene beads with a diameter between 1500 and 3000 nanometers, which precludes sizing applications. However, from the interlaboratory comparison study, I can confirm that for sizing submicrometer particles, this is generally no issue. The last disadvantage is that the current approach requires an extrapolation from the measurement of bright reference particles to dim EVs, which may affect the accuracy. This last problem emphasizes the need of reference materials with scattering properties similar to biological particles, which brings me to my final slide. In the European metrology project METVES2, we aim to develop a reference material to calibrate all aspects of vesicle flow cytometry, which are fluorescence, number concentration, and light scattering signals. In contrast to most currently available reference particles, the developed reference particles will have physical properties that resemble EVs. So keep an eye on our website. And thereby, I would like to thank my colleagues for the nice collaboration and my funding agencies for their financial support.
Hello again. I'm now going to show you the latest release of the FCM-Pass software. As well as performing light scatter calibration, this software allows you to perform fluorescence calibration and create automated MIFlowCyt-EV reports of your experiment with respect to the calibration and characterization fields. One of the distinct differences to the Xometry software is that this is free for academic use and is compatible with reference materials from any source. FCM-Pass catalogues your reference materials so that their information will automatically be completed in your MIFlowCyt-EV report, saving you time. You're also able to keep records of your calibrations and experiments over time. So today I want to take you through calibrating a dataset from start to finish with the FCM-Pass software. This is the new version 3 software. So these are some BD Quantibright MESF beads, and I'm just going to show you how they were gated. So we're gated on forward scatter and side scatter here. And we'll just look at the PE channel and turn this into a histogram. And you can see each of the gates for the beads. And just to do the same with the NIST traceable beads, this is the violet side scatter height. So we've used the height here because we're defining the trigger, whereas the fluorescence channel, we are using the area statistic. And for all of these populations, we're using the median. So we're going to add a data set now, a database. So this is the University of Ottawa's Cytoflex S, where this was generated. So we'll create a cytometer where we'll store this data set. We'll click on the cytometer, and now we'll add a data set. So these data sets are, this database is made so that you can track your data sets through time for each of your instruments, which will help you keep track of your experiments, but also your instrument sensitivity. So we've now created a data set. We just need to import our FCS files now. So we'll click the plus icon, navigate to where our files are and click open. So this will now import all of our FCS files, and it will also extract some of the parameters like the threshold for each of the channels, as well as what the parameter names are. So we're going to just define these sample types first. That will help with our annotation. So we're going to turn all of these NIST traceable beads into side scatter calibrators, and our fluorescent beads, our 8 peaks and our P quantibrights will turn into fluorescent calibrators, and the rest will just leave as standard samples. So now that that's done, we'll move on to adding a fluorescence calibration. So we can add as many of these as we like. So from our catalog, we'll pick that we want to perform a calibration using these PE-MESF beads. The quantibrights, you can see that their reference values have appeared on the right. We now select the channel that was relevant for those PE beads. And now we'll go to our data and put in the median statistics for each of the populations for the PE channel. And you can see that the parameter name there we've left as the default, which is PE-MESF, but you could also edit that. In the advanced settings, you can also change the FP ratios. So now that's all done, we'll go to the scatter calibration. We decide which channel we're calibrating, which is the violet side scatter height. You can see that the scatter threshold's automatically been detected. We'll change the wavelength, and you'll see that the sheath refractive index is updated. And now we pick the bead set that we use for calibration. So this is in our catalog. It's predefined with all of the metadata. And again, we go back to our FlowJo data, and we put in all of the median channel numbers for each of these bead populations. So by default in the software, if you were going to press calibrate now, it would automatically generate models for EVs with a low, average and high refractive index, which you'll see in the table here on the right. What we need to do is add one more refractive index in to represent the virus. So this was put as 1.4546, and in the FCS file, we want this to appear as MLV diameter. So we'll just edit that in there, and that will be added to the FCS file as well now. So that's all we need to do now for the calibration, and all we have to do is press calibrate. I've left this purposely in real time so you can get an impression of how quickly this can be done. And we've essentially gone from start to finish with this calibration now in less than 10 minutes. So when all of these files are calibrated, they'll be exported to the same folder the files were imported from. There'll be a folder in there called FCMPass export and the time that the files were exported, as you can see here on the right. And in there we've got our calibrated files. We also have our MIFlowCyt-EV spreadsheet. So this has been semi-completed now for the calibration parameters. The upper fields are for the metadata. All of the information that we could pull out of the FCS files in the catalog have been completed. So this sample acquisition info has all of your limits of detection across all of the different models you've generated. So polystyrene, silica, EVs, murine leukemia virus, and scattering cross-section. We know what the threshold was on every channel. The fluorescence calibration, we have all of the metadata, including the acquisition statistics, the reference values, slopes, intercepts, FP ratios, as well as the manufacturer information. And then finally, we have our scatter calibration. So this table just needs reformatting slightly if we make this first column smaller. But this table essentially, again, has all of the metadata associated with the model that was just generated. So we have all of the bead information in there, the refractive indices of the beads, the catalog numbers, the acquisition statistics, and everything else that you'd need to know to reproduce this model if you didn't have the FCS files. So the last output we'll look at is the quality control plots. The first page has the light scatter calibration model fits and your scatter diameter curve there down at the bottom. And the second page contains your fluorescence calibration plots. So this will just be a page full of the fluorescence regressions. So now just looking at the calibrated files, you can see when we go to the drop down with all the parameters, all of the standard parameters are at the bottom, and on the left you can see that the parameter that they were originally converted from. Something that I wanted to show you is that many of the scales might need changing. So the diameters, for example, we want to go to a linear scale, and ideally we want to look at the smaller range. Everything we're kind of interested in with this murine leukemia virus is down in the kind of 150 region. So if we put the scale to 100 and to 500 nanometers, it should be sufficient. And you can see here, if I just gate around the 152 nanometer beads and the 203 nanometer beads and get the median statistic for polystyrene, we come out at 148 and 197. So we're 4 or 5 nanometers out, but we're very close. And this is how the data was generated in the paper that was published. So you can see PEMESF versus murine leukemia virus diameter here. So I've now taken you from start to finish of performing fluorescence and light scatter calibration with the FCM-Pass software. I really hope this has convinced you how quick and easy it can be to perform calibration, as well as the huge benefits that there are to utilizing it. I've also put some references here to the papers that are relevant to the development and the implementation of the software, as well as our website link where you can download the software. So with that, I'd like to thank our collaborators and hand over to John Nolan.
Good morning or afternoon, wherever you may be. My name is John Nolan, and I will wrap up this tutorial by telling you about fluorescence-based EV size measurements, which we perform using vesicle flow cytometry. First, I'm at the Scintillon Institute in San Diego, where my group develops new technologies for cell and molecular analysis. We do most of our biology through collaboration with academic and industry groups. I'm also the founder of Salarcus Biosciences, which offers some of the tools I'm going to describe today as services and assay kits. What I'm going to tell you about today is I'm going to give you a little bit of background. I'm going to emphasize the role of instruments and assays in vesicle analysis, and I'll highlight again the guidelines that have been recently produced for reporting EV flow cytometry methods and results. Then I'll describe the vesicle flow cytometry, or VFC assay. I'll talk about characterizing the instrument in order to validate it for VFC. I'll describe our vesicle size standard, and then I'll describe and demonstrate how we use that to calibrate the surface area of EVs, which is how we estimate size. And then I'll wrap up by comparing and contrasting the different approaches to EV counting and sizing, and talk a little bit about how the integrated use of these methods enables rigorous and reproducible EV measurements. So first, when people ask me about how to measure vesicles, most often they focus on the instrument, which might be distinguished by its, lasers or detectors or collection optics that might make it more or less suitable for measuring EVs. But often what people fail to recognize is that the instrument is only a small part of the overall assay. And the results of an EV analysis will depend heavily on issues like sample staining, any wash steps, how you decide to use your flow cytometer in terms of a trigger channel or fluidics, and then obviously calibration, gating, and reporting all have a major impact on the results that are reported. And it's that realization that led to the development of this guidelines framework, where all of those aspects that contribute to the measurement of EVs, including the instrument, but also all of the other parts of the assay that are so important. So that little bit of background, let me describe for you our vesicle flow cytometry assay. It is a fluorescence-based assay that employs a fluorogenic membrane probe to selectively stain membrane particles. And together with properly validated fluorescent antibodies, enable the selective detection and immunophenotyping of individual vesicles. Calibration of both vesicle size and immunofluorescence are key to that, and I'll get to those next. In its commercial form, the vesicle flow cytometry assay, contains a detailed protocol for assays, which are all performed in 96 well plates. This is to encourage reproducibility. Likewise, the data analysis is very standardized. It takes place in FCS Express, which is a nice general purpose flow cytometry analysis software, and has the nice features also of being able to do parameter math, which is important for calibration. And it's also available in a reader format, which many people find useful if they don't have the full version of the program. So appropriate calibrators and standards are an important part of any assay. And in the VFC assay, we use several. We use a synthetic lipid vesicle that I'll describe in a little more detail as a vesicle size standard. It also serves as the negative control for immunofluorescence. We have bead-based fluorescence intensity standards and antibody binding standards that are useful for rigorous and reproducible fluorescence measurements. And then we use actual cell-derived EVs as appropriate, especially for immunofluorescence. So first, let's just say a couple words about instrument characterization, because all of assay performance will depend on a properly characterized and optimized instrument. And here, as for conventional flow cytometry, multicolor, multi-peak rainbow type beads are very useful. Your commonly available 8-peak rainbow beads will work. We've developed a set of 4 nano rainbow beads that are a little better tuned to the brightness range we'll encounter in EV analysis. And we use these beads to characterize several aspects of the instrument. We use the CV of the bright bead to report on laser alignment to make sure our lasers are well aligned across all lasers. The bright bead is also calibrated in terms of its concentration, so it serves as a counting bead for purposes of calibrating your sample flow rates. The performance on the individual fluorescence channels is evaluated by a separation index between the blank bead and the dim bead. And so for our membrane stain VF red, we've established a quality threshold of a separation index of at least 15. And for immunofluorescence, one can do a similar thing. So PE, which is probably the best, brightest immunofluorescence stain for immunofluorescence of EVs, we can set a threshold there. These fluorescence beads can be cross-calibrated against MESF standards so that we can report our brightnesses in those values. And what we can have here is just a little case study of one new instrument that a collaborator received. And when we ran the beads, we noticed that the alignment all looked pretty good. The separation on our membrane stained VF red channel was fine, but the performance on some of the other immunofluorescence channels on the other lasers was not up to snuff. And this let us flag this issue right away before we spent a bunch of time trying to measure vesicles. We had a service call, tuned up the instrument, and were able to evaluate the improvement in performance across the channels and know that we have an instrument that's ready to go. And so that's an important first step in setting up an EV assay. I mentioned cross-calibration. That can be done by using some of the commercially available MESF standards to cross-calibrate rainbow beads in MESF units. And those would be suitable for use on your specific instrument that you perform the cross-calibration on. So now we'll turn to the vesicle size standard and its use in surface area calibration. So as I mentioned, we have a synthetic lipid vesicle we call Lipo 100. It's of a defined lipid composition that's similar to an average mammalian cell. It's been extruded through a nucleopore membrane so that it has a relatively uniform size distribution. Notably, it has lipids in it, but no proteins. We can take this liposome preparation and characterize it by orthogonal methods such as NTA and RPS. And then we can stain and measure those vesicles by flow cytometry. And the way we use that to estimate the size of our vesicles from their fluorescence is we take the fact that we know what the size distribution of the liposome is because we measured it by one of these orthogonal methods. If we know the diameter, we can calculate the surface area. And if we stain that liposome with VF red, we can measure its fluorescence distribution. So we have a range of vesicles here that range in size from somewhere less than 75 nanometers out to perhaps as high as 200 nanometers in this case. And if we perform a linear regression of these two distributions, the surface area distribution from NTA and the fluorescence distribution from VFC, we find a straight line. And the slope of this line can be interpreted as the number of fluorescence units per nanometer square of surface area. So we use this linear regression then to calculate the surface area of vesicles based on its fluorescence measurement, and then use geometry to calculate the diameter distribution. And so it's through this relationship between surface area and vesicle fluorescence that we derive our vesicle size estimate. And in practice, this is done via FCS Express's parameter math, where we can develop a calibration equation that will take the VF red fluorescence, use the slope and intercept from that linear regression to calculate an estimated surface area, and then use just geometry to calculate the diameter. And so that's part of the analysis layout for VFC that lets us present the vesicles in terms of their estimated diameter and go on and measure immunofluorescence. The size estimates by VFC are quite concordant with these orthogonal methods. So here's an example of some 293T EVs from culture media, which have either been unfiltered or filtered and measured by RPS or VFC. You can see the size distributions. You can see a dilution series to show that that size distribution doesn't really depend on the concentration. And if one looks closely, one can see slightly different size distributions based on whether these have been filtered or not through a 0.22 micron filter. So the size estimates are concordant with those other methods. And once you've done that, you can go through and do the rest of the essential controls, which include the dilution series I showed before, a detergent treatment to show that what you've detected really are vesicles. We can move on and start doing immunofluorescence using the appropriate positive and negative controls. A few words on light scatter. So light scatter is a very useful parameter. We use fluorescence to estimate size, as I mentioned, but we notice distinct differences in light scatter intensities amongst vesicles from different sources that are similar in size. And so, specifically in this example, we're measuring the light scatter intensity of our Lipo 100 vesicles as well as some platelet-derived and red cell-derived EVs. We find quite striking differences in light scatter, which is most likely due to the refractive index and cargo that those vesicles carry. So wrapping up here, we find that VF red fluorescence is proportional to membrane surface area, and that gives us a means to estimate vesicle size from fluorescence. We use the LiPo 100 standard, which has been characterized by orthogonal methods, and lets us determine the slope of fluorescent units per nanometer squared. VFC has the advantage that it's specific for membrane particles, including lipoproteins, which could either be a challenge or an opportunity. And finally, the diameter estimates we obtain are comparable with those other methods across vesicle types. So what are some of the limitations? Well, so we rely on those orthogonal methods to estimate our size distribution of vesicles. And those have their own limitations, which I'll refer to in a minute. We're estimating really surface area, membrane surface area. And so it's possible that if you have some specific EV types, and in our experience, viruses and virus-like particles are a couple of these, they have very high protein contents. They will tend to size a little bit small by this method because they are so loaded with protein. In those cases, one would simply use a preparation of virus or VLPs as the size standard. Lipoproteins are also detected, but as we, I think, heard earlier, these can often be distinguished by their distinctive light scatter. And finally, the instrument sensitivity does matter. If you can't detect the standard, it's going to be very difficult to detect, to estimate the size. A couple words on these orthogonal methods. Nanoparticle tracking analysis and resistive pulse spectroscopy are widely used. They're both useful tools in their context. They both provide label-free estimates of vesicle size, diameter in the case of NTA, volume in the case of RPS. But they're both non-specific. They'll detect any particle. They have their own limitations in terms of sensitivity and dynamic range. They don't really have a way to measure cargo by themselves. So they tend to work reasonably well in purified samples, but as sample complexity increases, they tend to be less useful. So just to summarize these various methods, they all have their strengths and weaknesses. NTA and RPS, in our experience, are useful for characterizing purified standard preparations, but less so for complex samples. Flow cytometry-based light scatter is useful, and the label-free estimation of size and refractive index are attractive, but they have the downsides of it's a pretty non-specific measurement, and the math is not simple, although it's made simpler by the software packages we just heard about. VFC has the advantage of being membrane specific, way to measure surface area, and it's formulated for high throughput analysis. Again, it depends on this external standard, which relies on those orthogonal methods, which have their own limitations. So to wrap up, counting and sizing individual nanoparticles is challenging. Counting and sizing EVs is even more challenging, but we've seen several approaches whereby flow cytometry can estimate EV size and refractive index, number and concentration, which is a subject on its own, as is the measurement of molecular cargo. Perhaps we'll touch on those in future tutorials. Common features of EV flow cytometry are a optimized calibrated instrument, appropriate standards, and appropriate protocols that are applied rigorously. Looking ahead, we can anticipate that there will be new flow cytometry instruments which will have improved sensitivity and resolution. We know that people are working on better and brighter dyes. And so both of these anticipated improvements are going to make our lives easier. And then finally, we can look forward to integrating the light scatter and fluorescence measurements I described here to develop EV refractive index as a useful parameter, which I think is going to be useful. So with that, I'll stop. Thank you for your time and look forward to your questions.